Thanks again for the invite. I'm really excited to get to share my research, some of my research I've been doing here at UW with everyone. So today we're gonna focus on exploring variation in relative abundances of harbor seals in the Salish Sea. And just before I jump into the talk, I wanted to recognize my collaborators, Scott Pearson, Marty Raphael, and Beth Gardner, and then acknowledge our funding source, the Puget Sound Partnership. All right, so hopefully you're all seeing my first slide. And I guess just somebody shout if it didn't change. But assuming you can all see it, harbor seals are a really useful animal for indicating ecosystem health, just like most other marine mammals. And that's because of their life history traits of being a pretty long-lived species. They're slow to mature, so females generally will reach maturity at about age 3, and then males will reach maturity a little later at about 6 years of age. And also their reproduction is fairly small. Females will produce no more than 1 pup per year, and generally they're not producing pups every single year. Harbor seals are also a generalist predator. They feed on a diversity of prey items that include different species of cephalopods and also multiple fish taxa. They've also been described as central place foragers. So what that means is the animal will have sort of a home base that they come to in between all of their foraging trips. So for harbor seals, that home base is what we call a haulout. And a haulout is just an area that's out of the water. It could be a beach, it could be a little island, it might just be a rocky outcropping that might not even be available at all tidal stages. So it might simply be only available at low tide. But basically it's a place for seals to get out of the water, and they use their haulouts for resting. They use them for pupping and nursing during the breeding season, and also for thermoregulation. And so because these haulout sites are so important for harbor seals, anytime they leave to go forage, they're going to often come back to these haulout sites for those life history activities that I just mentioned. And so being able to predict spatial behavior can be really a useful tool because it can allow us to manage an ecosystem better. It can allow for spatial planning of various human activities, and it can allow for an understanding of how some ecosystem functions occur, especially when we're mapping out the spatial behavior of a predator. And so the spatial behavior of individuals is often dictated by foraging, kind of avoiding becoming somebody's forage, and also different breeding opportunities. So given that, we would not expect the individuals to be uniformly distributed across the landscape or the seascape, as sort of illustrated in this image here. Instead, we're more likely to see individuals clumped. So if we use UW's campus as an example, we would expect people to congregate at academic buildings, at restaurants, maybe at some recreational facilities. And then if we take a snapshot of this in time, we might also capture some people traveling between these points of interest. But what if we didn't really know what the points of interest were for humans? Say we're an alien species and we're coming into Seattle and we want to observe human spatial behavior at UW's campus. And we see different buildings, but we don't know what they mean. If we come in and we collect some data on these humans, we can see, oh, okay, they're clustering at these different areas, and we can infer from those data, oh, okay, perhaps these restaurants, these academic buildings, these recreational facilities are really useful to humans. And then if we want to take that information and apply it to another college campus, we would be able to use that information to see, oh, okay, this campus also has academic buildings, it also has restaurants, it also has recreational facilities. We're going to predict that the humans are going to be congregating in those areas. So that's where we're going to try to put in our efforts and expect to find them. So that's useful. But what if those places are also moving? So for example, if instead of going to a restaurant that's in a location that's, unchanging, what if the humans had to chase down a moving food truck that was traveling on an unpredictable route? That would make it a lot more difficult to predict where people are, especially at lunchtime. And that's sort of exactly the scenario that we find predators in. So predators are chasing down mobile prey. It can be useful to use predator locations to try and locate prey, or I'm sorry, the flip of that, it can be useful to try to use prey locations to locate where the predators might be, but it can be difficult if the prey are also moving. And so we are very fortunate, though, that a lot of environmental variables are going to be affecting prey in a fairly similar pattern. So instead of trying to use prey locations or variation in prey to try to determine where predators are, instead we can use variation in environmental covariates to try to predict where predators might be. And then to complicate things just a little bit more, if we think about harbor seals as generalist predators, they're not just going after one prey source, they're going after a lot of prey sources. And so that means that they have more options in terms of where they might be looking for their prey, because different prey items might specialize in different areas, and their abundances might change, which might just have cause the harbor seals to shift to a different prey source. But luckily, like I said, most of the environmental factors are going to be affecting the prey species in a fairly similar way, or at least in a predictable pattern, so that those might be a little bit more useful to search for predators versus just using that connection between the prey. So harbor seals are found in a number of places all over the globe, but one of those places is right here in our backyard in the Salish Sea. The Salish Sea is an incredibly diverse and productive system. It is home to more than 250 fish species, about 172 bird species, 37 mammal species, and all sorts of other taxa that are not listed here. And so being able to understand the spatial patterns of harbor seals in the Salish Sea can be really useful for managing this ecosystem that has just a great biological, cultural, and economic value. So the objectives of this research was that we employed hierarchical distance sampling methods to estimate the variation in relative abundances of harbor seals, to identify areas of high and low relative abundances of harbor seals, and to identify the environmental covariates associated with these relative abundances of harbor seals. And so for the distance sampling field techniques, we used boat-based surveys, and we used line transect surveys. And so we have about 100 areas to choose from. So the scope of reference is what we call a primary sampling unit, abbreviated to PSU. And what a PSU is in this case is about 20 kilometers of water that's adjacent to a coastline. And each PSU is adjacent to the next one, so there are no gaps in between them. And I know it's a little bit difficult to see from this map, but there are about 100 PSUs in here. They're outlined by the orange frames here, and I know some of the southern ones kind of all blend together. But from these 100 PSUs, 30 of them were selected for sampling during the summer and winter seasons, and then an additional 19 of those, plus those same 30, were selected for sampling during the fall and winter surveys. And so the PSUs that were selected are indicated by those purple stars here. And then if we break down a primary sampling unit or a PSU a little bit further, there is an inshore subunit and an offshore subunit. And so for the inshore subunit, it's right against the shore, and there are different routes that go out 100, 200, 300, and 400 meters from the shore that were selected for each survey. And each of those lines is further broken into a 5-kilometer segment. So within one PSU are 4 5-kilometer segments that run end to end. And then for the offshore subunit, these were traveled in sort of a zigzag pattern to try to cover more of the water, since it's a bigger area there. So the offshore subunit was 1.5 kilometers from the shore, and those zigzags were between 15 and 20 kilometers long. And so for these surveys, 2 observers would be aboard the vessel, and each observer would search out in about a 90-degree angle from their front to their side of the boat, depending on which side they were on. And they're just constantly searching for wildlife. And when they would see a harbor seal, they would record that they saw the harbor seal, and they would record the perpendicular distance that the harbor seal is to the boat. And so that sort of leads us to the concept of distance sampling. So distance sampling is a really robust method. It's really time efficient and cost efficient compared to other methods used for estimating abundance, such as mark recapture. And part of that is because you can actually gain a lot of information just based on one survey alone. And so the central tenet of distance sampling is if you have a transect, in this case we use the line transect method. So if you're traveling along a line, you should be able to see all of the animals that are right near you. But as you look a little bit farther away, those animals become more difficult to see, and the probability that you will just miss it altogether is increased. And so the concept of distance sampling is to estimate abundance from the product of the number of animals counted and the study area divided by the product of the area sampled and detection. So right now we're sort of focusing in on that detection piece. And the way that we can help estimate detection is that we can break those different distances away from the transect line into equally sized distance bins. And then we can take the number of animals in those distance bins and set them up as a histogram. So here's an example histogram that was taken from all of the surveys from 2009 across all PSUs sampled for that year. And so on our x-axis, we have the perpendicular distance away from the transect line. And then on the y-axis, we have the number of observations. And so as shown in this histogram, the first bar there, which represents the shortest distance to the transect line, so basically right on the transect line, that's where the greatest number of harbor seals were detected. And then moving farther away to a final distance of 275 meters away from the transect line, there are much, much fewer harbor seals that were detected there. But the concept with this decreasing detection with an increase in perpendicular distance from the transect line is that in this small strip of transect, we would expect that there's a fairly uniform distribution of animals. Now, I don't mean to contradict what I said earlier about the animals not being uniformly distributed across the seascape. That is true, but we're just taking a very small slice of seascape and about 550 meter width. We should expect to see a pretty uniform distribution of animals. So inferring from our transect line, there were about 155 harbor seals sighted for this year, for 2009. And then if you follow that dotted line out from that first bar, it would predict that even at that distance of 275 meters away from the transect line, we should also expect to see roughly 155 harbor seals rather than the 45 or so that were actually detected. So we can use that imperfect detection to help figure out an accurate estimate for what our abundance of harbor seals is. So the general idea here is that the detection is calculated from taking the area under the curve and dividing it by the area of the rectangle. And by rectangle, I mean the area enclosed by the dashed lines on the plot to your left. And so we can use different key functions to set up the curve of this plot. A more popular one is the half normal key function, and that's the one that we used for this project. And just like the name sounds, it is half of the normal curve. And so that is explained by this equation here. So just looking at the right side of the equation, we're taking the exponent of our distance, our perpendicular distance from the transect line indexed by k, which is just the distance bin. So was it 0 to 27 meters away from the transect line, or was it farther away from that? We're going to square it, and then we're going to divide that by the product of 2 and sigma squared. And so sigma can be further defined using some covariates, which we did. And then I just want to note real quickly before moving on that we indexed each of our sigmas. Again, that's just the shape parameter of the curve here. That was indexed by J, which is the PSU segment. And so there are some challenges with detection. Some of them we can model, and some of them we can't. So for example, when we're studying harbor seals, even if there is a seal right on the transect line, it could be missed because it might not be available for detection, meaning in this case that the seal is simply underwater and it remained underwater for the entire time that the boat passed through, leaving the observers unable to detect that animal. But other things we can model. So we did that in our model looking at Beaufort sea state. So for anyone who's more terrestrially inclined, that's just a measure of essentially how wavy it is. So a Beaufort sea state of 0 means that the water is extremely smooth, and a Beaufort sea state of 3 or 4 means that there are a fair number of waves that might obstruct your view of an animal like a harbor seal. And then we also looked at a random effect of the observer pair that was on the boat. So not every survey had the same observers recording harbor seals. And so there might have been some variation in the ability for different observer pairs to detect harbor seals. And so if we take a look at the detection component of our model described in the equation on this slide, we are separating out the Beaufort sea state into 4 different categories. So the intercept in this equation is a Beaufort sea state of 0, and then BSS 1, 2, and 3 indicate sea states of 1, 2, and 3. And then we have our random effective observer. All of these are indexed by J, which again is the PSU segment. And each of our parameters was given a vague prior. And then the other component of the model is the biological process. So for this part, we are estimating the abundance of harbor seals. That's indicated by N. And then we modeled this using a negative binomial distribution, and that's because our count data were over dispersed. And so the parameters for the negative binomial are lambda for the mean abundance, and then R is our dispersion factor, and then these were indexed by transect distance bin. So that was sort of what I showed in those histograms, and then again, J is that PSU segment. And then if we take a look at defining lambda, we included a large number of environmental covariates. Those are all indicated by the betas, and we'll go through those in more detail in the coming slides. But if you look at the last line of this big equation here, those parameters are indicating our random effects. So we have a random effect of year, a random effect of the PSU segment, and then we also accounted for the area that was sampled because not all segments of the PSUs were the exact same area. And again, we used big priors for all of our parameters. So if we take a look at just the spatial environmental covariates, we looked at the Haulout Site. We included distance to main river mouth. So that's an in-water distance to the main river mouths. We looked at depth of the water and the categorical distance from where harbor seal was seen to, or where the boat was, to the shoreline. And so I say it's categorical because that was either, was it on a nearshore subunit of the PSU, or was it on that offshore one? And then the last spatial covariate was looking at the different shoreline type. So thinking about the Haulout Site, the way that we included this in the model was that we took a 30-kilometer buffer around each haulout, and that was selected because 30 kilometers should capture the effect of the haulout. So that can include social interactions of harbor seals, and it can include the bottom resting, and also there's a lot of variation in studies that track how far from a haulout site harbor seals tend to forage, but 30 kilometers is sort of, it's a pretty conservative estimate. Harbor seals can forage more than 100 kilometers away, but they might not be as tightly associated with those haulout sites. And so the way that we put these into the model was that we looked at the proportion of a 30-kilometer buffer around a haulout site within a segment of a PSU, because we had multiple haulout sites in some PSUs, and a couple PSUs didn't have any. So the way that we quantified it was just taking the proportion of the PSU segment that had haulout area in it. For shoreline type, harbor seals tend to select different shoreline types for hauling out. So they could be found on a rocky beach or a sandy beach, or, just some rocky outcroppings, as I mentioned earlier. And so in 2002, some people working for NOAA went out and they defined all of the shoreline types in the Salish Sea in an effort to better understand how potential oil spill mitigation might occur. But it actually turns out that this could be really useful for predicting harbor seal spatial behavior as well. And so in a study site in Alaska, Montgomery et al. 2007 found that harbor seals were very, they were selecting for rocky shoreline types far more than any other shoreline type. And so it's possible that the harbor seals in the Salish Sea might have a similar affinity for this rocky shoreline type. The in-water distance to river mouth was included because we thought that since harbor seals sometimes feed on salmonid fishes, the river mouth might be a great place for harbor seals to come in and forage, because of the life history of salmonid fishes moving between the saltwater and freshwater. So we predicted that this might be influential on where seals occur. And then if we move on to our spatio-temporal covariates, we looked at the sea surface temperature, we looked at salinity, and we looked at chlorophyll A, which is a proxy for primary production or primary productivity, which kind of scales up to how many fish might be available for harbor seals to eat. So we collected data from the MODIS database for the chlorophyll A and for sea surface temperature, and then salinity was downloaded from the HICOM database. And then all of this information was downloaded through the Google Earth Engine platform, and then I ran some Spacio temporal interpolations to match the information that was available, because we didn't have complete spatial and temporal data available, so used interpolations to match the values for these three covariates to each PSU segment on the day that it was surveyed. And so our thought process on these covariates was that we thought harbor seals might be found in greater abundances at sort of intermediate temperatures, at somewhat lower salinities, just because some of their prey seem to prefer lower salinity areas, and then at spots where, or places and times where primary productivity is high, as indicated by high levels of chlorophyll A. And then for our temporal covariates, we looked at the upwelling index, which in the Salish Sea, upwelling is not as strong of a factor as it is off on the coast. The Salish Sea, most of those inland waters are sort of tidally dominated, but there are some effects of upwelling, especially in the Strait of Juan de Fuca and a little bit moving further inland towards parts of the northern parts of the Puget Sound. So we thought upwelling could potentially impact prey species and in turn impact locations of harbor seals. We looked at the North Pacific gyre oscillation, which is just a climate index. So looking at a large scale of how sort of El Nino and La Nina effects or events can affect the northern Pacific waters. And then we looked at what's called the fall and spring transition dates. And these just describe the dates at which the dominant species in plankton communities change. So those change seasonally, and the differences in those dominant species in those communities can affect the relative abundances of different species of fish, which can then affect where harbor seals might expend foraging energy. And then our final temporal covariate was looking at just whether it was during the breeding/molting season, or if it was not the breeding or the molting season. And so the breeding season for harbor seals occurs in late May, early June, up through August. And then the molting season just follows that. So could start in August and then go through September. And then this slide is really just a sampling to show you that our model converged and had good fit. So the top plots, density and trace plots, are just a selection of some of the covariate or some of the parameters, just to demonstrate that our model converged. And then we had Bayesian p-value for detection of 0.75 and a Bayesian p-value for the biological process component of the model at 0.78. So that indicates that we had good model fit. So hopefully you will believe my results in the next few slides, given that information. So we'll start with the detection component. And we're going to focus on the random effect of the observer pair. It turns out that that seemed to have a greater effect on detection than Beaufort Sea State. So if we look at our first plot here, the x-axis is showing average, above average, and below average detection for observers. So again, as a random effect, we're just looking at what the variation in the detection ability was. And then the y-axis is showing the estimated detection for the observer pairs. And so there was a fair bit of variation based on who was doing the detecting or the observing. So our average observing was around 0.55 or 55%. And then the above average observers were right around 70%, and then the below average observers were right around 40%. And this is for Beaufort sea state of 0. Again, that's just very smooth water, not much wave action to obstruct your view. But this very similar looking plot was based on a Beaufort sea state of 2, so getting some wave action in there. And as is shown in this plot, the estimated detection by the observer pairs is very, very similar to that first plot showing the Beaufort sea state of 0. And then if we think about the biological process, we first looked at the effects over time. So our x-axis is showing our years, and then the y-axis shows the estimated relative density, and that is to the PSU segment. So the area is 5 kilometers long by 550 meters wide. And as this graph shows, there really was very little variation across years. Then when we looked at the effects of different covariates, we had some negative effects from a couple of our different shoreline types. So sheltered tidal flats, boulder rubble, both had a negative relationship with harbor seal relative abundance. And then the distance to river, whether or not it was near shore or offshore. So in this case, the relationship was that the near shore seals, or in near shore areas, seals were more abundant. So those are our negative relationships. And then our positive relationships are the sheltered rocky shores. We're more likely to have greater abundances of harbor seals. Same with human-built sheltered riprap. And then we had positive relationships between Haulout Site and harbor seal relative abundance, as well as depth and relative abundance. And so we went through and we figured out our predictive density maps based on what our model was providing us. And we found some evidence of hot spots and cold spots. So hot spots and cold spots were calculated based on the coefficients from our model and looking at 5 representative days for all 20 years of all of the PSU segments that were sampled. And or ones we also predicted for ones that were not sampled. And we took the top 75th percentile and above as hotspots. So that meant that for all 5 of those representative days through all of those years, the density had to be in the top 75th percentile or greater. And then for identifying a cold spot, very similar technique. We just, instead of the 75th percentile, we looked at the pixels that were in the 25th percentile and lower, and those were recognized as a cold spot. And so on this map, the pixels represent a 2 kilometer by 2 kilometer square, and the density as represented by the color bar or color gradient shows that many seals to be predicted in each 2 kilometer by 2 kilometer pixel. And then we also calculated our uncertainty using the 95th percentile or 95th percent credible interval width. And so this map is showing that we had pretty tight estimates, not a whole lot of uncertainty about them. And so for the top plots, that's showing where the seals were in the breeding and molting season. And then the bottom plot here in C shows the relative abundance of seals in the non-breeding and non-molting portion of the year. And so the map is showing a lot more of the yellowish color, which indicates greater densities. And that is most likely because when seals are not breeding and not molting, they're more likely to be in the water. They're spending less of their time hauled out. And so they're going to show up in the surveys more frequently. And here is the associated uncertainty with the non-breeding and non-molting season. And again, pretty tight estimates. Just a few of these were a little bit wider. And then if you take your attention to where the hotspots are located between A and C, they're pretty similar. So based on the time of the year, whether it's breeding molting time or not breeding not molting time, the hotspots really didn't change a whole lot. The only difference is that there is one additional hotspot that lit up during the non-breeding and non-molting season. And if we zoom in a little bit more on those hotspots, the San Juan Islands had a number of hotspots in this area, and we think a big reason for that is because there are a lot of Haulout sites in there, and some that are slightly bigger. So the size of those circles that indicate Haulout sites indicates the relative size of the haulout. So just a ton of Haulout sites in the San Juan Islands. Another reason or another potential reason for high relative abundance of harbor seals in the San Juans could be that this was the only place in the entire Salish Sea that was dominated by sheltered rocky shorelines. And again, going back to that Montgomery et al. Paper from 2007, we have some evidence that at least some of the same species really seem to prefer these sheltered rocky shorelines. So that could be a big draw for the harbor seals to come in to this area. It might relate to the prey perhaps prefer the rocky shorelines as well, but that will be parsed out in a future study from, well, and then another kind of hotspot area is in the Admiral. I believe there were 2 hotspots in this area. Again, a number of large haul-out sites were found in this area. This area is very dominated by a lot of mixing from freshwater inputs and saltwater inputs. Just given its location, it's a high energy area, and it has also been identified by Olson et al. In 2018 as a hotspot for resident killer whale foraging. So that could be indicating that there's some pretty good prey here that harbor seals might also be interested in. And then our last area that lit up as hotspots was in the Kilcene Bay area. And this one was a little less obvious to us as to why this was a hotspot for harbor seals. If we go to our Haulout Site covariate, which seemed to explain our other sites, doesn't really explain a whole lot here. There are a couple Haulout sites, but not as many as some of the other areas, and they're not huge. So one idea could be that in Kilcene Bay there are some large river mouths nearby, most notably the large Kilcene River, and this could be a nice feeding area for harbor seals. Also in this area, just adjacent on the land, is a large dedicated wilderness area, so that might keep noise and just general human disturbance pretty low in this area, and that might be something that harbor seals are seeking out. And then the last idea that we had is that there's a national fish hatchery in this area, and that might be providing some prey species for harbor seals to come into this area and really make a good living here. So from our hierarchical distance modeling adventure, we were able to discover that the spatial covariates were the best predictors of harbor seal relative abundance. It was very interesting to us that all of the covariates that came out as being important, which we defined as not overlapping 0 in their credible 95% credible interval, they were all spatial covariates. We found that the harbor seal relative abundance varied really very minimally across the years. There was a little bit of a difference between breeding, molting, and non-breeding, non-molting seasons. It did not come out as an important covariate, as in its 95% credible interval did overlap 0, but very slightly the 88% credibility or credible interval did not overlap 0, and we did see some differences in those predicted density surfaces. So there seems to be a little bit of an effect there, even if it's not a very large effect. And finally, harbor seal are possibly buffering against some species-specific prey availability. That could be why we have a number of hotspots. If you were quick on the draw and noticed on some of those maps, some of those hotspots were also pretty close to some of the cold spots. That could simply be indicating that harbor seals are very mobile and they are kind of chasing prey species as they become more available, more abundant, and you know, they're not clustering into as predictable of locations as I showed in our human example with UW's campus earlier. And with that, I will take any questions.
Thanks so much, Jamie. That was fantastic. I really liked the UW campus analogy. Brielle, do you want to start maybe with questions from the in-person audience, and then I can take questions from online or something like that?
1 second, I will ask people if they have questions. Mark has a question. Mark was wondering if you do counts at the Haulout site.
I'm sorry, can you repeat the question?
Did you do counts at the Haulout site?
Oh, no. So these counts were all based on in water sightings of harbor seals. If there were harbor seals hauled out that observers could see, they did not include those in their counts.
Okay, thank you. That's our last question for, our only question right now that we have, but I'll unmute myself if we have any more.
Okay, and I'll just give folks a second to type questions if they have them. You can also, if you're online, you can raise your hand and I can unmute you if you'd prefer that.
I have a quick question, Jamie, while we're waiting for questions to come in. It's real. I was wondering, since you've got a whole suite of covariates that you added to your model, were there any additional ones that you didn't add in your model that you wish you could have?
Yes, that's a good question. So, as I'm sure many in the audience noticed, we did not include any top-down covariates. We only had sort of bottom-up effects. And we would have loved to include some top-down stuff. So the transient killer whales will consume harbor seals. So they are not, harbor seals are not apex predators in this system. They're mesopredators. And also, so resident killer whales and then also stellar sea lions will predate on harbor seals as well. And we were really hoping to include those in our models because the same surveys that went out to count harbor seals also counted those two species, among others. But unfortunately, the sightings from both of those species were just too low to actually be meaningful enough to include. So we did not include any effects of predators. We didn't include any effects of human disturbance. We just couldn't find good data for that, which of course doesn't mean it doesn't exist. We just weren't able to include it in our models in the time frame given. And then the last thing that we wanted to include, or I guess we did include, but we had to make a decision about, was what kind of climate index to include. So, you know, there are different climate indices. There's the PDO, which is like a Pacific decadal oscillation, and then there's the ENSO, which is the something southern oscillation. Sorry, my brain's not quite clicked in on that today. But we had to select just one of those climate indices, and so the NPGO, or the North Pacific gyre oscillation, seemed to have the greatest effect on species in this area. So that's the one that we selected for this one. But yeah, we did have a little bit more thought going into some covariates that did not make it into the model.
Oh, thank you.
I've got two questions for you, Jamie. The first is from Beth, who asks, do you feel like you can comment on what future harbor seal abundances might look like based on your results?
Sure. So I think I'll answer this in two ways. So based on just addressing just sort of overall harbor seal abundance, this population, or actually what's in the Salish Sea, is, I guess, three populations, or they're managed as three separate stocks currently in the Washington inshore waters. Those numbers are basically at their carrying capacity, and they are pretty stable. Those are based on some papers authored by Jeffries et al. And so I would think in the future, if things don't change a whole lot, harbor seal numbers will hopefully continue to stay about where they are. And then I guess I would also answer in terms of relative abundance and where to find a greater densities of harbor seals. I would think that projecting into the future, with maybe some different, or I guess just some changes to potential fisheries. I think that the fisheries industry might have some effect on harbor seal local abundance, or relative abundance, just because they are competing for the same resource, but not always, because harbor seals are generalist predators and they can always switch to something else if there is too much competition. But yeah, I would think as possibly fishing boats move around, that might shift where harbor seals are spending more of their time. But yeah, based on our spatial covariates coming out as being the most important predictors, you know, if that tends to stay the same, I would think that there's probably not going to be a whole lot of change in their relative abundance through future years.
Great, thanks, Jamie. Another question from Carrie Akala, who asks, did you look at interactive effects among covariates? She says she might have missed something, but it looked like you were assessing each covariate individually.
Yes, that's correct. We just looked at main effects. We did not do any interactions among covariates. I think our model was fairly complex enough, and so we did give it some thought, but we thought that might have been a little too much complexity to introduce into this model, and really we're just it was more exploratory, so we're really just trying to see, you know, out of a number of things that we thought could be related to relative abundance of harbor seals, you know, which of these are really coming out as being important, and then maybe some future work could dig into some interactive effects.
Yeah, understood. These models are a beast. I'm not seeing any other questions come in, and we're a little bit over time, so thanks everybody for your attendance. The bummer about being virtual, Jamie, is you can't see us giving you a round of applause. That was a really fantastic talk, and thanks so much for coming.
Yeah, thanks again for the invite. I feel very honored to get to come and do this, and sorry I couldn't be there in person.
Next time. All right, thanks again, everybody. Please join us next week. We've got an in-person seminar next week. It's a joint seminar from Tim Essington and Chelsea Wood, and it should be a good one. So see you next Friday.